Okay, so we're very happy to have with us Dr. Luc Teyton, MD, PhD at Scripps Research in La Jolla, California. He is going to be speaking to us regarding his newspaper, A Vascular-Associated Fibroblastic Cell Controls Pancreatic Islet Immunity. And very interestingly, the title of his talk is How to Understand the Initiation of Type 1 Diabetes. A short bio on him. Dr. Teyton received his medical training in France and specialized in rheumatology and trained in clinical immunology. He practiced medicine for 10 plus years, followed by a PhD in immunology at the Pasteur Institute. As a fellow at TSRI, he determined the dual role of MHC class II associated invariant chain. His research is focused on the T cell receptor and the molecular mechanisms of T cell activation. His group has made important contributions in the structure and function of MHC class II molecules, the presentation and recognition of lipids by the immune system, and the mechanism that leads to autoimmunity. I'm going to drop the link to the paper in the chat, so you'll have that at your fingertips. And please feel free to add questions into the chat. We'll also take questions at the very end. Thank you so much, Luke, for joining us. This is really a fascinating topic, and we're really interested to hear more about the paper.
Thank you for the invitation. The front is going to be it's not philosophical, but it's really, after 30 years in the field of type 1 diabetes, kind of my thoughts about the disease, on where we should be going. So
I love that. Yeah. And it's not just the paper, it's really sort of a larger impression and understanding of where we are now.
Yeah.
Thank you.
See if I could move the slides, it would be better. There we go. So the field, since I've been doing type 1 diabetes work, the obsession has been the cure. And we've had advocacy group for type 1, which is JDRF, on the like of it, saying we need to find a cure. And we've been obsessed with that idea that we should get a cure. I think we've been a little too quick to respond to that request. Again, coming from the medical field, in medicine, most of the time, unless, again, you get to very complicated situation, the thinking is always linear and binary. We've lived on the postulate of Core, that's why I put him up. A disease has a single cause, and once you have identified the cause, you can cure, and then you can move on. So again, remember, you have to identify the microbe. In the case of the core postulate, you have to isolate and grow that microbe. Then you have to induce the disease in healthy people, and then you have to re-isolate the microbe from the infected people. But that goes for any type of medicine. It doesn't need to be infectious. It could be the same for a coronary that is blocked by a plaque. It's the plaque that is causing the symptoms. You remove the plaque, the symptoms are gone. And if you block the coronary of an healthy person, you're going to have the same symptoms. So one disease, one agent. And we've lived on that, and we've been obsessed with it. So remember the early studies done in Norway, on all Nordic countries, where we're trying to find the trigger of type 1 in people who have genetic susceptibility. We removed cow milk. We did a bunch of things with all insulin, trying to induce tolerance on. And then we are still looking for a single virus that could cause the disease, or a single bacterium that could cause the disease, or a single type of food that will induce and trigger type 1. The issue is, if you look at everything across autoimmunity, we've never been in that situation. We've never satisfied the core paradigm by having the idea that there's a single cause for anything. So maybe we're making progress between EBV and multiple sclerosis recently. But again, even there, if EBV plays a role in triggering the disease, it's way more complex than just a single type of EBV. Because you could argue a lot of people are susceptible to the disease. There's a lot of EBV infection, but only few people get the disease. So I think type 1 on any other type of autoimmunity is going to face exactly the same type of situation. So again, it's not that we love to ignore the complexity, but it's easy to ignore the complexity. It's much easier to find one agent and deal with it, you know. So again, I'm going back to medicine, peptic ulcer for the stomach. Yeah, sure. There's one bacteria that is causing most of them, but it's not the only one. But in that case, you could say that Helicobacter pylori is clearly causing ulcers and is associated to transformation into cancer at some point. We are not in that situation. So the other thing that is, again, when you move to human studies, the human population is genetically amazingly polymorphic. So in type 1, we're not lucky, it's the situation. You know, the complexity of the genetic is dominated by a single feature that is a single mutation on the beta chain of HLA-DQ.
Have a good day, Max.
Excuse me? Oh, I thought that someone was asking a question. So,
Sorry.
but there's, I think someone is trying to ask a question or.
No, it's okay. I just muted the background noise and someone
Okay,
in
no worries.
a meeting.
The polymorphism in the population has to be looked at differently. Is that, you know, where we are coming for type 1 is that most likely it's the intersection of particular genetic susceptibility with particular environmental factors that matters. So it's two complexities on top of each other. On the next slide, that's what I'm trying to get to. At the bottom, I mentioned that, you know, the good German studies that have been done in young population with COVID-19, we've seen a definite increase of the incidence or the triggering of type 1 diabetes on the increase of about 2 to 3% in that population that they've studied. The same way, if you look at the meta-analysis of all the rotavirus vaccination, one of them said the diminution of type 1 in that population was 13%. So again, it's a high number. It's probably not that high, but there's clearly an incidence of, you know, vaccinating against a single virus on seeing an incidence in the general population on the incidence of type 1 diabetes. The same could be said about the microbiome. There are probably bugs that are protective, some that are causal. And then we end up in a situation where, you know, we make observation and we think that we've put the finger on what is essential, but it's essential for only a subpopulation. So to me, my thinking about type 1 has been, again, dominated by complexity. That's all I wanted to say. That is probably 100 ways of getting to type 1 if you have a susceptible genetic background. And I'm saying that because, again, if we think more generally speaking, why do we have, and it's the exception, it's the only endocrine organ we have that is not a single organ. It's distributed in 1 million structures in all the species we know with islets. So even in fish, when you have the appearance of the pancreas on endocrine function of that organ, islets are already, you know, islets. So there's got to be a very, very strong evolutionary reason why instead of having a single organ, we have multiple mini organs. That to me should absolutely dominate our thinking about type 1. And the other thing that dominates my thinking, there are actually two lines. One of them coming from that effort we made 30 years ago of transplanting islets. So there were 10 centers that were harvesting islets from donors on, if you split the people between 18 to 45, on 45 to 65, you realize that the older people have about 50% the number of islets of the young people. So it's got nothing to do with the harvesting technique. It's got to do with the fact that all of us are losing islets over time, which again, if you think of any endocrine function, is completely, exceptional. The thyroid will not do it, the adrenal will not do it. None of your endocrine functions are going to have that attrition over time. So that's very, very unique. So the question is, in many ways, do we all have type 1 diabetes? And then I go back to the fact that we have 1 million islets. If you think of it, if you have 1 million, you can lose a few, you're going to be okay. If you start the attack against a single organ, now you're in trouble. So again, that's where the 1 million islets is making sense. So in any case, that's the framework to think about type 1. On the other reason to do the work we do in my lab, on how I got there is I felt that in many ways we're kind of stuck. And if we don't understand the islet immunology at the very basic level, we will never make progress in understanding type 1. So let's look at the normal islet. What conclusion can we draw from looking at the normal islet in understanding the autoimmunity against that organ? So we've published 3 papers that on my computer I called 1, 2, and 3 that were exactly trying to do that. Can we look at the islet in the NOD mouse or in human, find the commonality of those islets, and understand how the immune system will get to that organ and attack the organ? So that's a paper we published in JI a year ago. The first one, a good read of the literature, was saying that there is no lymphatic drainage of the islet. So we've confirmed that in the mouse and human. The live one staining is the staining for lymphatics, and you don't find lymphatics associated to the islets, or at least not at that level of detection. So in many respects, again, like the eye or the brains, the islet is, immune protected by isolation. The second thing we noticed when we are doing, again, mouse and human work, we realized that there is construction of lymphatic structures over time, but they happen on the outside of the islet. So it's pretty easy to see on those slides. Where's my pointer?
I can see it.
No, but I just, there we go. Oops. No, I still don't.
I can see it over there in figure A.
Yeah. You see my,
Yes, I see
okay,
it.
thank
Yes.
you.
Yep.
You see that the islet making cells are not infiltrated. So that's my big fight in that paper to say, we've been talking about infiltration for decades. There's no infiltration, the islet is surrounded by tissue, and those are CD4 T cells. But on the outside of that CD4 T cell, what contains the CD4 T cell is lymphatic tissue, which you see again in the panel C. Those red cells, which are all CD4 T cells, are now in that lymphatic pocket that is drained by lymphatic vessel. And the same is in figure D. So the conclusion here is, there is no normal lymphatic structure draining the islet. There is no infiltration, and the construction of a lymphatic tissue is outside of the islet. And now I'm stuck. Ah. Sorry about that.
That's okay.
Let me kill that, or maybe, yeah, I'm moving now.
There it is.
Some observation in human. If you look at the human pathology, here it's a single patient from NPOD 6414, where we have all stages of evolution of the disease in that patient. And we make two observations. One of them, again, the CD4 T cells are red in that big panel. They are not infiltrating the islet, they are outside of it. And next to it is lymphatic vessels. You can look through that pancreas and you're going to find everything you would find in a mouse. You're going to see normal islets with glucagon and insulin making cells. You're going to start seeing some islets that have lymphatic drainage. And then you see less of the insulin secretion. And then you have islets that are completely devoid of any type of insulin making. So, same observation in mouse and human. The lesion is outside. The lymphatic tissue comes after the initiation, and the CD4 are sitting outside. So, if you want to see lymphangiogenesis, there's only one thing we know that creates lymphangiogenesis, or at least two. One of them is development. And during development, you have a lot of neo-lymphangiogenesis. And then at steady state in adult organism, the only way to get to neo-lymphangiogenesis is to have inflammation. So there has to be some inflammation close to the islet, creating the support for lymphangiogenesis on the incoming T cells. Is that spontaneous because of the location? Again, if you think of the pancreas, it's in a very dangerous place in many ways. It's encased in the duodenum, in, again, every species getting type 1. There could be a lot of things coming from the duodenum going and accessing the pancreas. Is that causal? Is that supporting the inflammation? What is really the nature of that communication between duodenum and pancreas? So we've sliced quite a few pancreata, probably 200, maybe 300, of node mice on mice that cannot get diabetes, like the single mutant beta 57, looking for signs of inflammation in the vasculature. If you look carefully enough, you will find signs of inflammation in any mouse you look at. Again, here it's the node on the mutant node. The blue is CD29, integrin beta 1 that goes up on the vasculature upon inflammation. Don't go look at the one that are all blue here. Those are exocrine pancreas. But here is a blood vessel in that left panel that is stained by MHC class 2 on CD29. So there is inflammation in that part of the vasculature. If you look at a non-diabetic mouse, which is that mutant, again, very close to the pancreas, and those are draining venules, you have CD29 expression, meaning that there's some degree of inflammation of that vasculature. If you look in the node more carefully, you can find in an islet that has not been infiltrated or attacked, has no insulitis yet, you have all the signs of inflammation of the vasculature next to it. You have nitric oxide synthase that is made by the endothelial cells. You have class 2 expression on, again, beta 1 expression. So it's kind of spotty in the pancreas. Some islets in their vicinity are seeing some inflammation. So what would cause the inflammation? You would think instantly of things coming from the duodenum going into the pancreas, and we've looked at the two obvious byproducts of bacterial life. Can we find MDP? Can we find LPS close to the islet in the exocrine pancreas? And the answer is yes. So again, you have 4 panels in A and B where we look at MDP, for which we've made an antibody, on LPS, for which there are many commercial antibodies. You find LPS on MDP in the pancreas. And if you look for the sensor of MDP, which is node 2, those are two successive sections in C. You find node 2 expression exactly where you find MDP by staining. So there are things coming from the pancreas that can be small molecules like MDP or LPS, and the sensors associated to it are also present, and it's always in the vasculature. So then we did some other type of experiment to see, can anything cross the duodenum and access the pancreas, exocrine or endocrine? We don't really care. The top panel, we are looking at lipids. So we gavage lipids to the mouse, and then 60 minutes, 6 hours later, we look if we find that fluorescence, the fluorescence of the lipid we've gavaged with in the pancreas, and we do. It's in the exocrine pancreas, it's in blood vessels, it's in the lymph node associated to the pancreas. If we do the same experiment with DNA, again, DNA can access the pancreas from the duodenum. And if we look at the RNA, same business. So you can use poly-IC, you can use CPG, you can use anything that has been labeled, you will find it in the pancreas. How does it get there? So we've looked at the vasculature in that panel D. You're in the duodenum, and you see that you can find the ovalbumin associated to the lymphatic structure of the duodenum. But if you look in the pancreas, in the overlay here, you have one artery where you have plenty of the ovalbumin crossing the arterial barrier, going all the way to the intima. But you also, you also have the staining of a vein next to it by the green color as well. So you find those things that are supposedly going through the duodenum, accessing the pancreas very freely. And then we did the experiment that to me is very key. If we gavage now one of those compounds that we know has pro-inflammatory capacity, can we increase the inflammation in the pancreas? And the answer is yes. So we gavage poly-IC to mice for 6 consecutive days, and we look for MHC class II expression in blood vessels that are associated to the islets. And you see that you have the appearance of that red color next to the islet. And all of those are blood vessels that are stained by the MHC class II, that is the best indicator of inflammation. You can do that in a mouse that is not susceptible to diabetes, that same mutant mouse I was talking about, the beta 57. You can do it in a humanized mouse that is expressing HLA-DQ8. And again, you gavage with poly-IC, you accelerate the disease, but in that process, the first mechanistic step that you can put your finger on, you get vascular inflammation. So that took us to the next logical step. We see inflammation of the vasculature. Is the vasculature very critical for what's going on? What we see is all outside of the islet. It's got nothing to do with the islet itself. That, by the way, is only blood vessels that are capillaries. In and around the islet, we have microvessels, but you are not talking capillary, and the nature of that vasculature is very, very different. So the order of things, we're going to look and try to look at antigen presentation, characterize the antigen presentation, and see what could be going on. Which, of course, took me back to more reading, more discussion with a lot of people I was interacting with at the time. If we go back to the original paper of Bottazzo, 40 years ago, when he published his founding idea that type 1 was associated to CD4 T cells on HLA class II, that's the image that is published in New England. The T cells labeled by an anti-CD45 are all sitting on the outside of the islet. And when you look at the HLA-DR staining, it's also on the outside of the islet, not within the islet. So that to me was clearly a huge point that was made, that again was reinforcing what we'd seen in the mouse on human, like he did. is that things were going on the outside of the islet. So systematically, again, off of the 200 or 300 pancreata that we sliced, we looked for MHC class II expression outside of the islet, not just within the islet. In the islet, we confirmed the work of Emil Unanue. They are macrophage, In section, you can see 2 or 3. There are probably a dozen of those cells per islet. But the majority of the MHC class II staining in mouse and human is outside. And it's outside and associated to the vasculature. So what are those cells? If we look a little closer, we realize that really in close connection to the islet, the red staining of inflammation or the red staining of MHC class II expression is associated to the basement membrane. So what is sitting on the outside of the basement membrane? So the basement membrane, it's not just that little membrane isolating the islet and making an islet. It's also the support for the structural feature of an islet, which is the vasculature. So that's a blow-up of that same image. Coming as at each of those junctions or coming out are going to be blood vessels. So again, we go back to historical images. It's kind of critical to remember that, again, overall, the vascularization of an islet is 1 or 2 microarteries coming in on 5 to 7 venules coming out. And they come out of that little mushroom of capillaries. On that, again, because I got through some good teaching when I was young, reminded me of one thing. George Pallady was given the Nobel Prize for his work on endothelial cells. And he's the guy who made that statement in some obscure paper that in tissues, the inflammation is always red at only one location, which is the postcapillary endothelium. And why is it the location of inflammation? It's because that's the only place where the endothelium is fenestrated. And upon endothelium inflammation, the fenestration increases and those cells can go through it. Why? Because it's the site of low pressure. It's the site where you find those windows to get cells through. So there's plenty of reason to think that for the islet, it's going to be the same phenomenon. If you have inflammation in the vasculature, arterial inflammation is exceptional, but it's going to be red like it's supposed to be red, and that's a postcapillary venule. So is that where we find class II? Yes, that's where we find class II. And we're lucky because in 2025, we have one antibody against a molecule called DARK that is a postcapillary venule marker that was identified, 10 years ago. So do we find class II expression there? Yes, we do. And that's where we find the signs of inflammation. And that's also the place where we're going to find that lymphatic structure on the CD4 T cells. So the site of inflammation of the islet that becomes sick is the postcapillary venule. And it's usually not a single location, it's usually multiple of them for a single islet. So, now we've kind of repositioned the disease not within the islet, but outside of it. And we did an amazingly simple experiment. Can we just use islet that we don't dissociate and stimulate T cells that will be coming? Because, again, it's so simplistic that I'm almost scared of talking about that experiment, but we take islets, when we co-culture the islet, we did multiple experiments on multiple times. The co-culture is going to be a few hundred islets with CD4 T cells from a transgenic mouse, and we're going to look at the state of activation of those CD4 T cells when we stop the experiment. So, start of the experiment, those are naive T cells, and the naive T cells are exactly what you're supposed to see. They are not activated, they are not expressing any of the activation markers we know. They are dormant cells. When we put them with islets from nut mice, we get a full activation that is a Th1 activation. If we block with anti-class II antibodies, it disappears. If we use islets from mice that have no MHC class II expression, we also have no activation. So yes, clearly activation of CD4 T cells when you expose them to full islets. So that was interesting. And that was kind of pushing us into studying those cells on the outside of the islet expressing MHC class II. And we did one more experiment. Can we isolate those cells outside associated to the vasculature? And can those cells spontaneously stimulate CD4 T cells? So the top panel, we look at week 6 and week 10 in nut mice. If we can find cells that are CD45 negative and expressing class II. And the answer is yes. Early on, at 4-6 weeks, we have no MHC class II expression by CD45 cells. If we come at 10 weeks, we've got plenty of those cells. We can divide those cells into two different families. One has endothelial markers, CD31, and the other one has just fibroblast markers, GP38. We sorted those two populations, and we put them in front of hybridomas to see if they spontaneously, without adding any peptide, would stimulate T cells, and they do. So they can stimulate. The right population is the fibroblast. The endothelial cell, that is the blue cell, cannot stimulate. So that was the end of that first paper, you know. We had located the MHC class II expression outside of the islet. There were at least two cells making that population. One was endothelial in nature, and one wasn't. It was a fibroblast. Only the fibroblast could stimulate. So a fibroblast outside of the islet, it's stimulating CD4 T cells. How would that be? So then I'm plugging into the body of work of Imolunanu again, you know. He showed clearly that there are peptides coming out of the islet that can access the circulation and stimulate T cells in the periphery. That was a Nature publication 5 years ago, where he clearly showed that that was going on, that many of the peptide byproducts of insulin synthesis end up producing peptides that are released at the time of insulin secretion. Here we show it, you know, we illustrate that idea visually by looking at C-peptide secretion from the islet. So the mouse is injected glucose, SAC 20 minutes later, and we stain for C-peptide, and the C-peptide is in red. And you see that you have that strange pattern where the C-peptide overlaps with the insulin secretion in green, but then you also have C-peptide by itself, and that C-peptide co-stains with the vasculature. So all of what is, again, that's a vein. So it tells you that at the time of insulin secretion, you're releasing C-peptide on other peptides that are the byproduct of the synthesis, and they access the vasculature. And we would make the argument that some of the peptides that we've observed, which are pretty much every peptide that has been reported as critical for type 1, we found being presented by those fibroblasts. Insulin is, of course, our main interest, but the BDC 2.5 reactivity is also found in those cells. So what are those cells in more details? Then that becomes very easy. You isolate by flow cytometry, and then you do SMART-6 sequencing, and you can tell what the cells are. That's the experiment in the mouse. You have a population of CD45 negative cells that are expressing class II, and those cells have those profile of expression. The only thing that should be noticed here, that's again, we confirm that one is endothelial in nature and the other one is a fibroblast. None of those are expressing co-receptors. None of those are expressing what you would expect from an antigen-presenting cell, which would be, the accessory molecule and the adhesion molecules that are critical to prime naive cells. They also express most of the time the B7 family members like PDL1. So we confirm two cell types. They are unique in the sense that they express, like you would expect from endothelial cells or fibroblasts that have been induced by gamma interferon, only class 2 and not very much else. The same is in human. It's exactly the same two population, expressing class 2 on being endothelial or fibroblastic in nature. And we can look at the expression of PDL1 here in a mouse, two successive sections because the two antibodies are from the same species, but the same cells that express class 2 also express PDL1. On the majority of the fibroblast, not the endothelial cell, but the fibroblast, when they express class 2, they also express PDL1, which would be the hallmark of gamma interferon stimulation. Gamma interferon induces those two molecules in a coordinate fashion in pretty much every cell type we know about. We've made a cell line off of it. Again, you have the endothelial population, you have the fibroblast population. We can separate those. We've made a cell line of the fibroblast population. We can stimulate with gamma interferon. They express PDL1 on MHC class 2. Upon stimulation, they resemble very much the primary cell. They modulate their collagen expression accordingly, like normal fibroblasts. And then we also showed two things that are very interesting. They are very phagocytic, but the endothelial cells are not phagocytic. So the only phagocytic cell of those two cell types expressing class 2 is only the fibroblast. They are good at presenting peptides. They are extremely poor at presenting protein because they are missing the real machinery of antigen presenting cells. So they present peptide, which takes me back to 3 slides ago, and they don't process protein very well. Can we confirm the existence of those cells by other means? We did it by using that, fucosylation technique. We put a fucosyl transferase at the surface of C4 cells, and we incubate C4 cells with islet that we've dissociated, and then by sequencing, we isolate the antigen-presenting cells and characterize those cells. Vast majority of them are fibroblasts. The remaining are B cells or myeloid cells that would be macrophage or dendritic cells. We've shown that for two antigenic systems, but the results are identical. So what it became really, that's going to be the connection to the follow-up papers that we are working on. We tried to look at the function of those cells. And the way we did it, we took anti-insulin C4 cells from transgenic mice. We pre-activated those cells for 10 days in vitro, and then we exposed them to myeloid cells or to the fibroblast that is expressing class 2. And again, keep in mind that's primary cells, primary isolation. The only cell that has been manipulated is the CD4 T cell. So the T cell alone, 10 days post-activation, are dormant cells. They have very little gene of activation that are expressed, as you would expect. You put them with myeloid cells, they go into a full Th1 phenotype. You put them next to islets that don't express class 2, nothing happens. You put them with that CD45 negative population that expresses class 2. Now you have a unique profile of genes that are genes of tolerance and naivete. You're back to the original naive CD4 T cells you were looking at. So you induce on pre-activated CD4 T cells a program of dormancy upon exposure to the fibroblast, which I think is really the critical factor on the critical conclusion of the paper that we've just published, is that not only those cells spontaneously present peptide, but their presentation in the context of no adhesion molecules on most often PD-L1 expression are capable of downplaying pre-activated CD4 T cells. To make it a bit more puzzling, we've looked at ES-derived islet that we are making by differentiation in vitro, and we've looked if we could find the equivalent cell, and we do. And we do also now, we have the same data for iPSCs. When you make those pseudo-islets in vitro, if you stimulate with gamma interferon, you're inducing MHC class II expressing cells on the outside of your pseudo-islets. And those cells look exactly, like the fibroblasts that you have isolated in primary islets. That's ES lane on lane 2. You have identity of genes between, what you've produced in vitro and what you see in primary cells. Again, the correlation coefficient for ES on 2 is about 80% for gene identity that we've looked at. So the conclusion is, looking at the basic immunology of islets, we've discovered a cell outside of the islet that is capable of sensing inflammation. And when it does, it's expressing MHC class II. And the function it has is that it can induce a tolerizing program in CD4 T cells. So we're not reaching the conclusion yet of saying that's critically important for going or not going into type 1, but we're providing the first element to look a little outside of the box on the idea we've had of the progression of type 1. We would say that cell is probably critical in normal condition when you don't have sustained inflammation, probably sufficient to turn down a CD4 that would come in and would show signs of activation. The other thing that to me is more puzzling is that we have outside of the islet that machinery on the ability of picking up peptides made in the islet, but picked up outside. So C-peptide is one, all the other byproducts of insulin or chromogranin or whatever you have is also picked up by those cells. What could be the function, and why do we keep that function in islet? To protect the islet. Why do we need to have those reservoirs of peptides in a cell type that is outside of the islet? Why isn't the macrophage in the islet taking care of those peptides? That to me is the big question. So we are doing, you can think, a lot of complementary and follow-up studies, knocking out class II in those fibroblasts, characterizing the population of CD45 cells outside of the islet, because I think there's a lot of regulatory function that we've kind of overlooked by not being careful enough on. It's not that we are not careful enough. We've been obsessed too much by what's inside of the islet. What's outside, clearly, for the regulation of islet physiology is critical, and we need to look at it more carefully. So that's the model that I kind of like to show. The postcapillary venule is really the regulator of the immunity of an islet. In normal situation, nothing happens. The CD4, the CD8, the dendritic cells are falling through that conduit. Nothing is going on. When you start having inflammatory cues that come from the duodenum, you open those little fenestrated endothelial cells, some cells come in, and they're probably downplayed by the fibroblast. And when you have sustained inflammation, you have sustained leakage, a response against antigen that are coming from the islet.
Luke, could you just say that one more time? You froze just for a minute there.
Where did I freeze? Between the inflammation and leakage on?
Yeah.
Okay. So yeah, normal situation, you're a non-susceptible individual. At time, you're going to get some inflammation, some leakage, and the fibroblast is capable of taking care of your few cells that go through. you're on a susceptible background, which allows you to sustain the inflammation or have cells that are more sensitive. Now you have more leakage and you have every professional immune cells coming in. Macrophage, dendritic cells, more CD4, more activation of CD4, more CD8 on. And now the fibroblasts cannot control that reaction anymore. And you have initiation of the autoimmune reaction. I'll finish by thanking everybody who's worked on it. So the work I've presented, 90%, it's the work of Don on Anne, a technician in the lab, on the postdoc in the lab, supported by a large number of people who've been in the lab over the last few years, many of them still working with us. And I also want to thank my two mentors in type 1. Now, the first one, people forgot about Hugh McDavid, but Hugh McDavid was critical in putting the HLA in the context of autoimmunity. And it's really you who told me work on type 1, because it's probably the easiest to understand for now. And the animal model is absolutely fantastic. So thanks to you. And thanks to Emil for the last 20 years before he passed, where we've interacted weekly, discussing those type of issues. Why do we have 1 million islets? Why do we have inflammation in the pancreas? What is that breaking point where we go between destroying a few islets and getting into type 1? And I think that without those discussions on those two mentors, I will not be doing the work I do. And I'm trying to, pick up the torch on the flame and keep working with the same ideas that those two guys had. On that note, I'll stop and take questions.
Thank you so much. This was just a fascinating talk, and I loved that you tapped onto the power of communication across career states and how important that is. And we find it very important at the Sugar Science too. We love that trainees and people that are just getting into the water with type 1 diabetes research can have access to all these great thoughts from KOLs like you. So thank you very much. Just like to open it up to questions. I have a couple. You're getting a lot of claps in the audience here. People are really excited about this talk. Would anyone like to unmute and ask a question or drop something in the chat?
If I may,
Yes.
this is Camillo Ricordi from University of Miami, former Washington University fellow, Professor Lessi, Paul Lessi, and Emilio Nano. I had the luck to work with them in the 80s. I'm pretty ignorant on your front. Those guys, surgeons transplanting islets and stuff like that. But I've been intrigued recently on the fact that when you find an NOD mouse that doesn't become diabetic and you get upset because you lose a mouse that you could use for a transplant experiment, you find that we went to look what is in that pancreas and all the small and the medium-sized islets disappear, and the normoglycemia is maintained by 3 or 4 big islets. And there is a population that I thought it rose an idea that in a population of cells that we always don't consider, in that incredible vascular structure inside the islets that you cannot understand how all the other cell can fit because it seems like a glomerular. There are a lot of pericytes that upon activation of inflammation or immunity, they can become MSC, start producing immunomodulatory counter response and maybe build an immunoregulatory response that is only successful in big islets, but is not sufficient in small islet after you have this activation by the perivascular fibroblast. And I was wondering if you think that could be a reason, because they should be all genetically similar in a mouse, but you see only the small one disappearing and not the big one. So I thought maybe it's an issue of failure of the bodyguards or enemy at the gate, that if you have a big islet, sometimes you can respond and other times you cannot.
So you're touching on 2 points that actually have made that same observation that you're talking about. We followed 45 mice for 2 years till they died, of good death. The one that we call non-diabetic, 45 non-diabetic mice over 2 years. And then halfway, we sack some of those mice and look at the pancreata like we did. And yes, you have islets that have been completely destroyed for beta cell presence, and some that are completely normal. So it tells you two things. One of them is, again, that we know from human studies, you don't need that much beta cell mass to sustain normal glycemia. The other thing it tells you, again, looking in very old mice, there is no sign of inflammation at that stage, no T cells near the islets. It's that the disease stops. So you can control the disease over time. And there are probably, again, some people out there who are in that same situation. They've had a bout of type 1, but they don't go into full clinical type 1. And I think, to me, that's the population with one antibody. Out of the people with one antibody, the vast majority never go to 2 or 3 on full type 1. But they keep that one anti-whatever, GAD or insulin antibody for the lifetime. And they are like that 20% of female mice that never get diabetic. So they've had diabetes and the diabetes stops, which in many ways makes you optimistic in maybe the ability of intervening early on stopping the disease in some way. You will not prevent the disease, but you could probably stop it. And instead of having 20% of female mice not diabetic, you may go to 80%. Again, I don't like the anti-CD3, but I think the anti-CD3 in many respects does that to some patients that have been treated. So again, that tells you about the evolution on the peaks and valleys that are probably the normal evolution in mice and human of type 1. It's not a single shot, a process that is ongoing that will never stop. Why would some islets be more susceptible than others? It could be their vascularization. Some of them may have 2 or 3 microarteries, a couple of venules that are close to a larger vessel. There might be more inflammation in that particular part of the pancreas or less. Some of those studies, are doable. It's difficult to do in a systematic fashion, but I think that the fact that the pancreas is constantly exposed to inflammatory cues will probabilistically give you those type of patterns of the disease and the pathology.
Yeah, that's fascinating. Thank you for the presentation.
Yeah, that was a great discussion. I just wanted to just tap quickly on the fact that Joanne Almaca at Alejandro, down in Miami, has brought to light the idea that these parasites, right, have a role in the pathology of type 1 diabetes, especially in the NOD mouse. And just thinking, and then sort of on the other hand, Sarah Richardson at Exeter has brought up this, or she discovered this small endocrine organ, right, that doesn't really have the extracellular matrix, and they disappear on the path to type 1 diabetes. So if you're thinking about, what you've brought up, Luc, this whole idea of the vascular, the extracellular matrix, and then we're talking about the periocytes and these small endocrine organs. How can those stories all coalesce, or can they? I mean, do you think it's a temporal hit, or is it just a perfect storm with all these different aspects being, impacted?
So kind of trying to take the Palladi post-capillary venule story into the 21st century, when you have rid of inflammation in those locations, what's happening at the cellular level? Again, we are immunologists, we've fractionated CD4 T cells in whatever number, probably 100 or 200 now. If you look at the vasculature, we are very naive. We think endothelial cells and then pericyte and not very much else. Every of those cells in particular location have a lot of diversity of cell population that are very critical.
Mhm.
So the pericyte the pericyte is essential because, again, there are myofibrils that can add to the contractility or expansion of that particular blood vessel in that particular location. So of course it's critical. Those are going to be exposed to inflammatory cues. Their function is going to change. They are very sensitive to nitric oxide, to increase in size. They are inducible by interferon gamma. They are probably the diversity of the endothelial population in those locations is probably completely overlooked for now. But we have the means of studying it. We can do single cell, and if we use SmartSeq, well, we can really get the full genome of that single cell. I think we'll get to a good dissection of those critical elements for inflammation on the immune reaction at the very front of immune reaction.
Here's a good question from Junbao Yang from Turn Biotech. Since during COVID, there's a 2 to 3% increase. Does that in type 1 diabetes? Does this support a cross-activation of anti-islet cells?
So again, I've nothing. There is cross-activation. Again, the term is very broad. In normal immune reaction, you get cross-activation by, the main way is cytokine. This cytokine-driven perpetuation of activation. But in the case of the interest of rotaviruses or COVID, those are enterotropic viruses. They infect the guts, they infect the pancreas, and they have the ability of infecting those organs. So to me, again, the study cannot be done, but I think it's really the infection of the organ itself or the inflammation it provides to the organ that is key in increasing the incidence of type 1 in that context. You add that one little thing on the scale, and now, boom, you tip the balance on going to clinical disease.
I guess I would just bring up one more thing is that, Molly Huber out of University of Florida and now at the Joslin brought up, or put out this paper recently, basically showing that beta cell dysfunction precedes insulitis and immune filtration. How would the VAF model accommodate that? Like, how does the VAF, is the VAF perturbation downstream? I mean, you've talked about the fact that it is not, but I mean, how are you gonna, encapsulate that work?
So the best experiment to position VAF in progression of disease is to knock out MHC class 2 in that cell, which we are doing now. We have to knock out one inducible, one constitutive by using the collagen genes. We've made 2 constructs to knock out class 2 expression in that particular cell type on every fibroblast. So again, if the prediction is true, if it's protective, if you remove the cell, you should have an acceleration of disease. Okay. Yeah. We shall see. We'll know next year. The mice both of them are made, so we know that. The other thing about the VAF that is of interest to me, there are 2 things. One of them I was telling you, most often it expresses PD-L1, but some of them don't express PD-L1. Again, if you go back to the basic of the molecular biology of gamma interferon induction, which is for now the only thing we know induces MHC class 2 expression in non-professional cells, the response element for PD-L1 on MHC class 2 are identical. So if you have cells that express only class 2, it means they've been induced by something else than interferon gamma. Yeah. And to back that up, you look at the phenotype of node mice that have no interferon gamma receptor, they still have the disease. The disease is delayed, but they still have the disease. So what is inducing MHC class 2 expression on VAF when you have no interferon gamma signaling? Yeah. There are a couple of ideas that are out there, but we've not proven any of them yet.
Okay, well, we're going to wait with very, really sort of bated breath for this new paper.
Monica, if I may Monica, it's great that you mentioned Joanna Lamaca because we just had a meeting last week and I've been bugging her on pericytes and their immune modulatory possible role because she's been studying the role on vasculature, more flow and stuff. And she finally agreed that if I give her another fellow to study immunology, we'll look also at this aspect. But if I can give you another piece of provocative information, we published, made the cover of eBiomedicine, a little paper showing if pericytes are immunomodulatory and become MSC immunoregulatory upon activation. We took islets from patient long-term type 2 diabetic that stop producing insulin, where the beta cell goes through this dedifferentiation or inflammation-based long-term. And we took these islets isolated and co-transplanted them with mesenchymal stem cells with MSC, both the islets in vivo and in vitro, and they went back to produce insulin like ages before. So it shows that maybe there is and I like this fact that I have an inflammation-centric view of the universe. So that I think that maybe both type 2 and type 1 diabetes has a deep root in common that is inflammation-based. And we've been spending our life studying the beta cells and the T cells, and is it suicide or homicides, but maybe it's the bodyguards that are failing in their
Yeah.
role. You know,
The protective, the
Yeah.
protective features.
Yeah.
Wow, this is just a great discussion. Thank you so much, Luc, for bringing this forward and the idea of getting some of the key opinion leaders in the room and talking this through and bringing these ideas together and connecting them across collaborative borders is very, very important. So thank you, Camillo, as well.
But thanks to you, you're making it happen. It's just your forum that you started, years ago, is proven to be amazingly useful. Yeah, I love those discussions as well.
Thanks, Luc.
Yeah.
We have everything 24/7 on demand for you,
Yeah.
all scientists and clinicians, and we're hoping and we hope we're being of assistance. So thanks again. We'll
Thank
talk
you.
soon. Looking forward to these new papers. Exciting times. Bye-bye.
Thank you.